Hey, welcome back. So we've been talking about how cells divide. So now I want to talk to you a little bit about the life cycle of a eukaryotic cell. It's not the entire life cycle of every eukaryotic cell, but it's basically how they get from birth to reproduction again. And it's a cycle we call the cell cycle, right? That's not too hard to remember. But the cell cycle also includes a component that gets pretty complicated. We're going to spend a lot of time on it. It's called mitosis. Okay? It's identical cell division, used for things like replacement and repair. It's going to be different from another one we'll talk about later. I'm just going to put it here. Meiosis. So that is the manufacture of sperm and egg cells. Okay? That is non-identical cell division. So that'll be different. Okay? So this is identical. And this is non-identical. Okay, well, let's get started. So let's go back to some basic ideas. Like, there's not only one type of cell in the world, right? There's prokaryotic cells. Remember, Okay, and those are things like bacteria, archaea, right? Stuff like that. But we are eukaryotic. Okay, so whereas bacteria and archaea they're teeny tiny and they have like, if you blew one of them up to see it closer, they'd have like a tiny circular chromosome and there's no nucleus and like maybe a couple of ribosomes. That's pretty much what they've got going on. It's very different from a eukaryotic cell. So a eukaryotic cell is huge by comparison, much larger than this. Okay, and we have a nucleus, and there's tons of linear DNA, like that's strands of DNA, and then we have bunches of organelles everywhere, and right, so we're very complex, we're very large. And so it means that if a bacteria and archaea are going to divide, remember when we looked at binary fission, it's a much simpler process. Make a copy of your chromosomes, split your ribosomes, and then split your cytoplasm and make two separate cells. Close it off with your membrane. Not too bad. But eukaryotic cells has a lot. We have a lot in there. We have to unpack quite a few organelles. Okay? And so part of this is also we have a ton of DNA. One of the things that allowed us to become so complex is every time we had gene duplicates, instead of just throwing them away like most prokaryotes did, sometimes we kept them and we changed them and we mutated them and then became more new functional genes, right? Allowing eukaryotes to become more complex, right? So we're complex. We also became larger because we had to house and organize and distribute all of these products, all right? We have more genes because we keep our duplicates and allow them to undergo different types of evolution in most cases, or a lot of cases. And because we have so many of these genes, we couldn't fit them all on that single circular chromosome anymore, right? We couldn't do that. So we had to make these like multiple chromosomes, and they're very large and they're all linear, right? That's the other thing. They're linear chromosomes within the nucleus. So each one of us eukaryotes has a characteristic number of chromosomes in each cell of the nucleus. It basically contains all of the information for making another copy of that animal. Or plant or fungus, or sorry, total eukaryotic mess up there. Right, so eukaryotic chromosomes are actually composed of chromatin. So that's an important idea. So we know about DNA, right? The DNA helix, so there's like a strand of DNA. But if you're going to move DNA, if you're going to move it into two new cells, there's some risk involved, especially with these really long eukaryotic chromosomes, right? A tiny little bacterial plasmid is going to be easier and we're at lower risk for breaks. But the thing is, these eukaryotic chromosomes are long. And if they are going to be pushed to one side of the cell and the other, as you see they are quite pushed around later on, they're at risk of becoming broken in places, right? All of a sudden you have fragments. And those fragments might be reattached in the wrong places or with the wrong DNA, introducing mutations, right? If they're even repaired at all. And that's not good. We want to kind of avoid that. Okay, so what we do, hold on, let's get a bigger eraser here. All right, so what we want to do is actually get a special set of proteins that are going to help us pack this up for later. Okay, and those proteins are called histones. Okay, here's a histone protein. Think of them as like cord winders or cord bundlers, right? And basically what happens is we wrap our DNA around it 2 times, right? And get like 2 bits of string around it, essentially. Kind of like that, and then they tether together. Histones are grouped in octamers, right? Hold on. Histones. Octamers. Right? Mer meaning of pieces, and octa meaning 8. So there's 8 of them altogether. Alright. So that's nice. So now we've cord bundled them up, we package them, they're really safe, it really protects them from getting nicked and broken and introducing mutations. So that's really what chromatin is, okay? So it's the DNA molecule and proteins that are going to help maintain that structure. They also do something kind of cool. They control the activity of the genes, okay? They do this by every time you have some parts of DNA that are wrapped around the histone, they're shut down, okay? When you have other areas that are open and exposed, it means that some of the machinery for transcription and translation can access this part of the genome. So it means it's on and can be activated, all right? It's one of the levels that we can control gene activity. There are many levels, as we'll talk about later in the semester, but that's just one to keep in mind over. So now we're preparing for cell division. And chromatin, because we've wrapped it around these histones, has become highly compact and it's thick. Now we can even see it with a microscope, which is always kind of fun. So before any of these eukaryotic cells can even divide, however, it's got to duplicate or make copies of all of its chromosomes. So we do 2 copies, right? Because cell division results in 2 cells, so we don't make more than that in this case, right? And so each one of these copies is called a sister chromatid. Essentially, it's like this. You have here, pretend like you just have a cell that has, 3 chromosomes. It's like it's a small eukaryote. It's got 3 chromosomes, right? So we have this eukaryote with 3 chromosomes. Now we want to undergo cell division. So what happens is we're going to make a couple of copies, right, of each one of those specific chromosomes. Alright, that's good. But now we've got these duplicates. Nice things are, we bundle them in the center at the centromere, okay, right here. Sometimes it's a little more acrocentric. Sometimes it's like more telocentric, meaning near the end, so it doesn't always have to be in the middle, but, in this case I'm putting them in the middle. Alright? So when you have them joined together in this twosome, they become sister chromatids, right? So they're chromatids, when you pair them together and they're of the same genetic material, of the same part of the genome, of the same chromosome, that's a sister chromatid. Alright, check. So, sister chromatids are joined together along their length. Yep. They are cinched especially tightly at their waist, right? So this is the centromere, and this is, a nice metacentric one, meaning it's like in the middle, right? And when a cell divides, these sister chromatids, they're going to separate from each other because the whole point was they're going to go into new cells anyway, right? So you have cell A and cell B starting to divide, right? And so that means that you'll have a copy of each one of the chromosomes for the new baby cells, right? So that's how that operates. Because you can't have a cell, a viable cell that's going to survive if it doesn't have a genome, a complete genome, right? Okay. So I know I just drew these gorgeous pictures for you guys, but let's take a minute and look at a better example, I think. Okay, so here's a single cell. It's got its single chromosome, right? Oh, here it is, just shown a different way. Just drawn so it looks more helicy, I guess. And so now this cell is like, I think I'm going to duplicate, right? So that's what this cell's thinking too. Yeah, let's make some copies. So the first thing it does is, so again, this is still that single cell. It makes copies. It makes one copy of each one of the chromosomes, okay? Those chromosomes, now that they're duplicated, are called sister chromatids for each chromosome, right? So they contain the same genetic material on that chromosome, and they're cinched together at the waist or the centromere, okay? So what happens is after cell division, we have 2 separate cells right? Two separate cells are born. Not born, just generated from cell division. Right? So now we have two separate cells. So if you look, they have the same number of chromosomes as their original parent. Okay? They didn't keep too many. That would not result in the best things for the cell's viability. Okay, I hope that clarifies things a little. So remember when I was talking about being able to see this with a microscope? Yeah, you can. They kind of, it's dense. It goes from being a string structure to all wadded up, plus it's got all the extra mass of just proteins in there. And so this is a pair of sister chromatids, right? Here's one on this side, right? Here's one. And then the other one's here on this side, right here, right? And they're joined together at the centromere. This is that little waist area that we were talking about. So it's kind of neat, right? Yeah, it's pretty. It's good. That's where you get that X look, too, right? You see a lot of chromosomes when we talk about cell division looking like this. Just know that that's a replicated or duplicated chromosome, right? And that this is a singleton, non-duplicated, just one. All right, so we've talked about some basic components that you need to know before we start talking about cell division in eukaryotes known as mitosis, right? So I think I talked to you a little bit earlier about this idea of the cell cycle, which is kind of like the life cycle of a cell. And not all of them go through all the stages the same way, but we're going to start looking at some of the sequence of events that they use in order to either complete replication or to join senescence, right? To just kind of exist without dividing, just being a cell. So there's a sequence of steps or events that lead us through the cell cycle. And so let's talk about what some of those are. There's two main big parts, right? So there's interphase, which is like the lion's share of the life of the cell, right? And then there's mitosis. So here's mitosis, and then everything else is interphase, okay? And interphase is generally divided up into about 3 different stages. And those stages, I intended that to be a circle, by the way, so if you could use your imagination, I'd really appreciate it, right? That's supposed to be a circle. Okay, anyway, so it's a cell cycle. My best representation. And in the cell cycle, we have interphase and we have mitotic phase. Okay, and that interphase is actually divided into a couple of stages, okay? And those stages are G1, S phase, and G2. And there's actually another stage. We're not really, it's not like on your test or anything, but I think it's important to know about. So hold on, let me draw these out. So here's G1, like S phase can live over here, and this is G2 phase. Okay, so G1 is just like, after the cells are born, so this is after cell division, right? So one of the cells, we're going to talk about the story of this one single cell right here, okay? It's born, it starts to grow, there's more cytoplasm, it gets bigger. It's just a toddler cell, and now it's got to get bigger. And so 2 things can happen. One, it can actually go into what we call a side, and it's called G0, which is kind of the side loop, and it's like a quiescent type senescent stage. It's just, basically, it's just not having babies. That is what it's just not dividing, right? But it's still doing stuff. It's still living or secreting or manufacturing or sending nervous impulses, like whatever it's doing, it's doing. So like yeast cells and neurons, they go into G0. S phase is where we start duplicating chromosomes, and it's actually synthesis phase. Look at it that way. Right? So synthesis, DNA, that's the major thing that happens then. So after we've made some DNA, we're like, well, we're really going to do this. We're really going to divide the cell now. So we go into G2 phase, which is actually just another growth phase. And so it's just getting larger, right? So it's big. It's got all that extra chromatin in there, right? Because it's duplicated. And it starts making a lot of like microtubules especially. There's just a lot of those. And we'll see why we're going to use those in a bit. M phase is the mitotic phase, right? So here's M phase right here. So that's the actual process of cells dividing. And the very last moment, or cytokinesis, is not really part of cell division, but it's that last minute when they push away from each other, right? And they become finely divided. The cytoplasm is complete, right? The membrane is completing at that point, and they move away and become individuals. So here's a much better circle. Right, so this is the cell cycle. So cell cycle. And I just want to say real quick, the cell cycle is not mitosis. The cell cycle is not mitosis. The cell cycle contains mitosis sometimes. Right down here, it's part of the cell cycle. It is a stage of the cell cycle. A lot of times people think the cell cycle is mitosis because we spend so much time going through the steps of mitosis. So I just say that a lot, and sometimes that helps people just remember, right? So here's gap phase. Here's our little teeny tiny toddler cell, and it's growing. It's getting bigger, more cytoplasm. We're just, some more organelles, some stuff like that. It can enter G0 over here, right, and complete any kind of like senescence. It just isn't dividing. It's still alive. It's still living. It's still doing cell stuff. It's just not going to divide. That's all that really is referring to. Here's the synthesis phase. You can see the nucleus here is darker because we're more concentrated, right? We also are bigger by this point. We've grown a bit. And then by the second gap phase, we're bigger. We still have that nucleus, but then also we're going to have a lot of these little microtubules that are starting to emerge. And that is one of the organelles that becomes really busy during the second gap phase. After that, we enter mitotic phase. So that's the actual act of cell division. There's a couple of steps to it, which we'll look at, and you'll have to be able to identify. And then finally, cytokinesis, the final division of the cytoplasm, the closure of the membrane, and the separation of the now two daughter cells. All right. Well, I hope that was helpful. If you have any questions, feel free to email me, and have a lovely, day. Normally I say night, but it's daytime today, so have a good day.